Greetings everyone, and today we are going to discuss about glyoxysome. Glyoxysome is basically a microbody just as peroxisome, and we can also call it as a specialized peroxisome. That specialized peroxisome is particularly found in the plant cells while it is absent in the animal cells. So it is basically of course originated from the Golgi body, and apart from this it is having its function to store store fatty acids material inside the plant cells, particularly inside the plant cells, particularly in germinating seeds, particularly in germinating seeds. So there are several plants. I mean, there is castor. Castor, its biological name Ricinus communis. After which there is cucumber. That is Cucumis sativus. After which there is peanut. Peanut is having arches, hypogee. After which there is corn, is having its biological name is Z-maize. After which there is soya bean. After which is having its biological name is glycine. So these are some biological names of these crops, and these crops basically contain their seeds. These are having seeds inside them, and these seeds basically are important. These seeds contain what? These contain endosperm. Contain endosperm, and within that endosperm, we know that endosperm is formed by the fertilization. During double fertilization occurs of the polar nuclei. When both of the polar nuclei on the opposite ends combine together, they form endosperm, which is a uh, food storing food storing triploid, which is a food storing triploid, which is a food storing triploid, which is a food storing triploid within the ovule, which provides nourishment to the embryo. Now, this food storing triploid is basically having food storing triploid. So here, this food storing triploid contains what? This contains glyoxisomes, in which the food is stored. And second function of this is that it converts the lipids into carbohydrates. Now, how does it convert? First, lipids are converted into fatty acids and glycerol, after which fatty acids are further converted into a 4 carbon sugar, which is known as succinate, and then succinate is converted into final product, which is glucose. Now, here in this process, when succinate, which is a 4 carbon sugar, which is not uh, part of, there's a formula as well. So this succinate is not the part of a carbohydrate. I mean that this is a sugar, but this complete process of making glucose was based on the conversion of non-carbohydrate compound into carbohydrate compound. So this process in which formation of carbohydrate takes place from non-carbohydrate compound is known as gluconeogenesis. Gluconeogenesis. Now, the process of gluconeogenesis occurs in such a manner that here um, first lipids converted into fatty acid and glycerol, fatty acid converted into succinate, and succinate is converted into glucose. Now, this occurs in a cyclic manner, which is known as dioxylate cycle. While this phase in which the succinate is converted into into glucose is known as gluconeogenesis. Is known as gluconeogenesis. Apart from which, this process takes place within the glyoxysome, in which we are having the crystalline core. This crystalline core basically contains the enzymes which convert the glyoxy basically it converts the lipids into the glucose through this process of glyoxylate cycle. And apart from this, this is a basic intro, and I guess that's what is present in our books as well. After which we are moving forward mitochondria. That's basic intro. That mitochondria, first of all, it is um, what's the size? Size is around 0.5 to 1 nanometer is its diameter. On the other hand side, it is 10 micrometer long. 10 micrometer long. Now, there can be multiple units of mitochondria within the cell, depending upon the and metabolic requirement of the cell. For suppose in heart cells, there are 350 mitochondria as compared to kidney cells, which are having less amount of mitochondria, around 50. So why mitochondria is more in heart cells? I mean 350, because in heart cells there's continuous contraction and relaxation, which is an active process, and that active process requires energy, which is created in mitochondria. Secondly, this mitochondria is having two parts. For suppose we know that mitochondria is having two layers, outer inner membrane. Now if the outer membrane is subtracted from mitochondria, then it is known as mitoplast, as it was in the case of cell. When we subtracted cell wall from the cell, it formed protoplast. In the same way, this is mitoplast, which is formed by subtraction of outer membrane from the mitochondria. After which, it is a self-replicative organelle, which means it can do self-replication, as well as it is having DNA, as well as it is having its own RNA, all three types of RNA, as well as it is having its own ribosome, but that is 70S ribosomes, as well as it is also having uh, enzymes, as well as it is having ATP synthase particles, ATP synthase particles. These are basically the F0, F1 particles, which we often call as Friedrich-Moller particles, which we are going to further discuss in the next video. But for now, these are a few things which are present inside the mitochondrial matrix. Um Here in this structure, we are having in such a manner that first we are having outer nuclear membrane, not the nuclear membrane, outer membrane of mitochondria, outer mitochondrial membrane, and then there's inner mitochondrial membrane, and the space between outer and inner mitochondrial membrane is on its intermembranous space, and inner mitochondrial membrane is having several folds inside it. It is having several folds inside it, which causes to form some sac-like structures, which are known as cristae. These foldings are known as cristae, which contains the following elements: DNA, RNA, ribosome, enzymes, ATP synthesis particles, F0, F1 particles, and there's of course cells in self-replication, as well as in the protein synthesis of mitochondria's own cell, on organelle, sorry. After which the outer membrane is having several protein channels. It is having protein channels inside it, which allows the permeability, of several several organic molecules which are coming inside the mitochondria. That is freely permeable, fully permeable. Uh, these protein channels are freely permeable, known as porins. But the inner membrane is partially permeable, selectively permeable, which allows the relevant amount of biomolecules, or the required amount of biomolecules which are required within the mitochondrial matrix. Apart from which, one more point here, that inner membrane is rough, outer membrane is smooth. Outer membrane is having, let me just first discuss the outer membrane. Outer membrane is having the ratio of protein to lipid as 2:1, which means that outer membrane is having ratio of protein to lipid as 2:1, which means that the number of protein is double the number of lipids. On the other side, in the inner mitochondrial membrane, the ratio of protein to lipids is 1:1, which means both are present in equal amount. Now this is a basic um, explanation about it, after which there are several structures such as there's double-stranded circular DNA present inside mitochondria. There's RNA, mRNA, tRNA, rRNA. But apart from this, we also call it a semi-autonomous organelle. After which we are gonna discuss something, a few things here as well. But apart from this, this mitochondrial matrix is having a DNA, which means it is able to self-replicate, as well as the ribosomes, which are present here, uh, produce protein, and this protein is only consumed by the mitochondria. And what purpose? In the electron transport chain, as we know that there are three stages of aerobic respiration. Glycolysis occurs in the cytoplasm, and Krebs cycle occurs in the mitochondrial matrix. All of the enzymes of Krebs cycles are present in mitochondrial matrix except succinate dehydrogenase, which is present inside the inner membranous structure. Let me just write it down. Succinate dehydrogenase, which is present inside the inner membrane of mitochondria. After which uh, we are having electron transport chain. Electron transport chain, that which takes place in the inner membrane, is having protein. Protein basically works as electron carriers. These proteins act as electron carriers during electron transport chain within the inner membrane of mitochondria, and finally cause to produce energy. And here, last but not the least, we are having examples. I mean, we can call that as ATP mill of cells, as well as we can call it as powerhouse of cell, powerhouse of cell. So these are a few things. Apart from this, rest of the things will be discussed in the next video, and hopefully you enjoyed the video today, and we are going to further discuss something